You and your mom.
It's beautiful. That's why I got the grant. Actually, not really.
Is that the make 3D picture or whatever? Yeah.
The whole reason for and actually I thought about making it so you can actually print them, like using a 3D printer.
Ryan's artistic side.
I've done trees in the past, but not this kind of tree yet, but in time. Anyway, so what we're going to talk about today is how phrapl works, and tomorrow I'm going to talk about whether it works. Right? So here is just sort of the background, the methods, and that sort of thing, and then tomorrow we'll talk about some of the results and empirical results that Nathan's run, and show that, like, yes, it actually can often give you the right model, and that sort of thing. Okay. And again, this is to educate you, so if you have questions, stop and I'll explain. Okay. So first, history, since we all care about history. So I worked as a grad student on a method to delimit species. Okay, and it was a non-parametric method that involved looking at structure within species and structure between species. And in the whole review process at SysBio, people started hitting me for not doing a parametric approach. It's like, okay, well, this is already an NP-complete problem. It's very hard for me to do a parametric approach too. Fine. So Knowles and Karstens had a model in 2007 where they used simulation, to estimate probabilities of gene topologies. Right. So I just extended that to the species delimitation approach, right, where I had different species trees and different assignments and used their simulation approach to estimate likelihoods. Okay. And that's for species delim. And then Brian and I started talking about the phylogeography situation again. Here's again a map of Middle Earth. I think we're the only NSF grant to ever include this as a figure in our grant. And so again, the question is, you have this region and you have stuff in the region, right? You want to know, what are the big phylogeographic things that matter? Is it, the Misty Mountains, or is it rivers, or that sort of thing? Okay. And what we do with phrapl is first lump these things. So let's say you imagine you have really good sampling. We first lump into populations. Okay. And right now you can do 3 to 4 populations. In theory, you can do more, but the complexity gets huge as you have more populations. Okay. But if you're willing to restrict the sets of models you look at, you can do that. Okay. And we don't have a good way yet to say how you should group into populations. Okay? You could use structure or something else, but that's not part of our method. Okay? So you have some set of populations, and now we can eliminate the map, okay? And sort of arrange them evenly. So then how can these populations be related with phylogeography? And what phrapl does is look at all the possible ways it can up to some limit for the amount of complexity. Okay. So, for example, you could just have populations coalescing. Okay. And there are many possible models that are just like that, right? That differ in time of when the coalescences happen and also the tree topology. Okay. There's different population sizes you can have, right? So this population here is a bit bigger size than its sister population. Okay. And of course, there's also migration. Okay, so you have migration between different populations. And so you can imagine this is getting to be very, very complex. And we actually don't even need to have a tree. So you can have some sort of, here's a classic island model, right? Stuff's dispersing. We can imagine something sort of in between, right? Where we have some sort of tree model, but then we just have some dispersal too. Okay? So there's a huge model space. All right? And then so what do you do with this model space? One thing we're inspired by is model test. Who's used model test? All right, and now there's like J model tests and successors, right? And this is a super popular program, right? And why? Well, what all it does is tell you, for your phylogenetic analysis, which of these, I think it's 72 models, fit your data best. Use that, okay? And it has, some heuristics built in. So rather than doing it, the proper way to do this is to estimate a separate species, a separate tree for every model. Okay, it doesn't do that. It just makes a quick neighbor joining tree, and fits that one tree to all these models. Okay, but it's a way of looking at this full set of models and quickly letting you say, okay, I'll use this, and then doing a more thorough analysis with that, right? So, does TRN plus gamma fit my data better? We use that. Okay. And so with phylogeography, there's not something like that. Right now, what you have to do is just say, I think it's this model or that model, right? Or this set of 8 models. Okay. And so what we want to do is have a system that will allow you to say, okay, I don't know anything about it. Tell me what the models are. Okay, that's one step of what we do. And this is a trend in phylogenetics. Right? So you have a lot of models that have a lot of problems that have specified hypotheses, right? So I think that this clade is evolving with this rate versus that rate, okay? Or I think I have a birth-death model or something like that. And then you can compare fits of different models, okay? There are other approaches that now will try a whole bunch of different models, right? So model test will say, okay, you want to try a whole bunch? We'll try all of them. Protest. For trait evolution, there's things like surface or Atur or Medusa, right? They'll try a whole bunch of models and then figure out which models fit best and draw conclusions from that, okay? And phrapl is in sort of that set, okay? And again, it can be used in a hypothesis testing framework, but it's also just sort of model testing, or if you want to be pejorative, data dredging, right? Fitting a lot of models. So we're thinking about that. Any questions about that framework? Okay. So why phrapl? Rolls off the tongue. Phylogenetic inference using approximated likelihoods. Okay, that's the name for it. The input are gene topologies. Okay, so what does that mean? So it means we don't care about branch lengths. We don't use branch lengths. We also don't use raw sequence data. You have to have some way of creating your gene trees to start. Okay, and so Nathan and Adriana have some sample scripts to let you run RaxML to get gene trees. Okay. You also need an assignment of samples to populations, right? So think about those first figures I showed you with a bunch of red dots and then yellow areas. You have to some assignment. You say, individual A, B, and C are in population 1. Okay, that predefined assignment file. Okay. Some of the jargon we use here: N is number of populations, okay, and K is number of free parameters. It's an AIC. Okay, because one way we limit the set of models is by saying, I want to use all models that have no more than 3 free parameters or 4 free parameters. Okay? And so the rationale here is that you have a certain amount of information in your data, right? So your data can only support a certain number of parameters, so let's limit it to those sets of models. Okay. So the overall high-level view of what phrapl does. You first generate all possible models, given n populations and fewer than or equal to k free parameters. Okay. And you see, here's a bunch of different kinds of models, right? So here we see, so these are all 3 population models, but this one has some migration and some population coalescence. This one has similar sort of thing, but also has a lineage that comes down after. We have, this model has just like an animal, which is just, gene flow between populations. So we generate all this set of models. Okay. And if you want to, you can take that set and bring it somewhere else. If you want to put it into IMA, you could. Right? If you want to do some ABC approach, you could. But this set is very scary and large, oftentimes. Okay. We can then filter models. Okay. And the filtering, there's various filters you can use, and you also develop your own. So you could say, I only want models that have no more than 1 migration rate parameter. Okay. Some of the models we have stored now are models that require symmetric migration between populations to make it simpler. Okay. If you allow free migration, it makes the model space a lot bigger. Okay. You can filter for only models that have a species tree. Okay. One practical thing to know about phrapl is that this step here is still slow. Okay. So we pre-compute some sets of models for you, right? So in the phrapl data directory, there's a set where for 3 populations, all models up to, is it 3 or 4 free parameters?
Through 3.
So they're already pre-computed, okay? And those took hours to run?
Yeah, well, right. There is one with 4, but for 3 populations, there's one where you have 3 free parameters, and then we have a 4 population one that only has 3 free parameters as well.
Talk a little bit about the free parameters and why.
Yeah, we'll get to that. Yeah. And this is something that we can improve in the future. Okay. So, and also think about if there's sorts of models you want, or sorts of filters you want. So you can say, okay, I don't really care about estimating 15 migration rates. 1 or 2 is fine. But I really care about estimating population size changing through time. And we can do that, but let us know. Okay, so part of this is to figure out what you guys want for your data sets. Okay. And also the filtering. First we do create all models and then filter. It'd be easier to filter as we create. And so there's speed ups there we can do. Okay. And after that, we analyze. So we can either find the best model for your data, or we can find for every model the AIC score and the AIC weight for the models. Okay. Sometimes, I mean, the sort of natural way is just find the best. Okay, here's the best model. But the next question you should ask is, well, how much better is it than this other one? Right? And to get at that, you can't just do a search. You have to look at several models. Okay, so what we'll be showing you the next couple days looks at all the models. Any questions so far? Okay. So step 1, generate all possible models. So one thing to think about is how populations can coalesce. Right, so I have 3 populations, what can happen? I could have them all having a polytomy, going to a common ancestor, right? Or I could have A with B coalescing, or B with C coalescing, or A with C coalescing. Or I could have no coalescence. Right, with an island model. Okay? And I could also create models where these go back in time and don't coalesce again, or models where they coalesce. All right, so here's all my possible coalescence models, right, for just 3 populations. Okay, and how does it affect number of free parameters? Right. Well, here I have this time, tau. When did they split? Right. And why does this matter? Well, it's matter if you think about if you have a population coalescence happening a long time ago, right? Then along each lineage, it probably had a lot of time for the gene trees to coalesce, right? What happened very recently might be a lot of incomplete lineage sorting because it has an effect on the topology we're looking at, right? And anything that has an effect on the topologies, we can then use the topologies to actually estimate that parameter in theory. Okay. So there we have 1 free parameter, 1 free parameter, 1 free parameter. Here, no parameters for this. There's no time coalescence. So for this, just the coalescence time, there's no parameters we're using. And here we have 2 parameters. Okay, you can sort of see how parameters are compounding. Now, within each little interval, we allow, in theory, different population size. Right? So here I have, on this particular model, 5 different segments. Right? So I could, in theory, have 5 different population sizes or 1 population size. And think about how this expands the model space. Right? So here, this always has population size n1. B could be the same as A or different. Right? So now I have 2 possible models. Right? AB could be the same as A or the same as B or a different one. Right? So now we have 2 times 3. So you can see how this expands. Right? C1. And note that we allow you to shift. So we have these sort of population coalescence intervals, right? So we allow n to change along those. Right? And again, okay. So we have all these different ways of assigning parameters, right? So if they're all set to be the same population size, one parameter, right? If they're all different ones, it's 5 parameters. And you can think about how this model space expands. Right, so I can assign them all to the same one. So n1, 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 right? Or I could do n1, n2, n1, n1, n1. The maximum model, so I have, this is a free parameter, this is a free parameter, and so forth. Right, which creates this whole tree of possible models. Okay, just for this one population history. Okay. And so if I say use this model, right, I have 1 collapse parameter, this time, and 2 population size parameters, this one and this one. Right? I can now start thinking about what that model means. Right? And for this first part, this first interval here, right, it means they have 3 populations, A, B, and C. Okay? And then A and C have the same population size, right? B is a different population size. And then I have a coalescence happening of populations, and then I have population A, B, and then C. Okay, so that's how you structure that model. And so if you look at phrapl, there's an object called migration array, and that has the full set of all the models we're looking at, and it's a list. And you can look at a particular element in that list, and that'll tell you something about the structure of the model. Okay, migration array, bracket, bracket, 17. The 17th model. You can look and see how we structure that. Okay, we'll see that when you do some examples. All right, migration. So far we've had no migration, but of course we need some migration, right? Because here, to have this all coalesce at one point for MS to run, there's some sort of way of getting gene copies from here over to here, right? And so here you have all these possible migration rates, and again, the model complexity becomes terrifying, right? So for this one arrow here, I can set that to be 0, no migration, or 1, migration parameter 1. Okay. For this next arrow, I can set it to be 0, no migration, equal to the first arrow, or a different parameter. Okay. And so forth, right? And so here's where, for the stuff we're doing during the workshop, we've set them to be, these two are always equal, but it could be 0. These two are always equal, but could be 0, so forth. Just simplify the model. But if you have a question where you say, I think what I have is, organisms going from A to B to C, but not backwards. Maybe they're going downriver or something. You can then fit that model. Okay, it's not in the current model set. Any questions about that? Okay. And so I can say, let this be my model. Right, let me look at the name of this model where I have migration from C to B in this interval, right? And I have migration between A, B, and C in this interval. And what we have is a way to visualize it, right? It's hard to see, this is a tree and this stacks up here, so you have a way where you can actually see it and get a sense of what this means. Okay? And if you have parameter estimates, it can scale the line thicknesses so it corresponds to parameter estimates. Okay? And those of you who don't have RGL installed, this won't run. Okay? Or if X11 is not installed, this won't run. Otherwise, you can create this sort of thing. Okay, and the color is automatic. You can change the labels if you want. Okay. And it doesn't have to spin. So for a paper, obviously, you just want the best view. So you can just drag it around until it looks pretty and then save it as a file. Okay. And again, part of the workshop is if there's something else you want for this, let us know. It's going to help you communicate. Because one thing that really helps people understand your biological question is to be able to visualize results. That's why structure is so popular. You can see oh yeah, okay, I have this red group and this blue group, and here's some group in between. It's that people just get it. Right? So we're helping people just get this. Okay. Yeah?
When you model population size changes, you model instantaneous population size changes, so there's no
Isn't
explanation?
that how it happens in reality? Boom. Yes. Right now we do. And one of the next steps actually is to allow you to have an exponential increase or a decrease, something like that.
Yeah.
Is that something that would be a priority for you for your data set?
No. I was just curious.
We'll put it off then.
Yeah.
Again, anything you add like that increases the complexity, increases the model space size. But if that's what your question is, then you could say, okay, I want to spend my compute effort on estimating that increase and decrease, and migration rates, I'll assume there's no migration or something, just one migration rate.
Mm-hmm. Okay.
So it's a trade-off that way. Yeah. Other questions about this? Yeah.
So when you're doing this initial step, do you have to set upper and lower bounds for each of your free parameters a priori? Would you put an upper and lower limit on population size, for example?
What this does is set this with the model structure. It's like saying HKY versus GTR.
Okay.
Right? And then later when you're doing the numerical optimization, then you can think about bounds for that. But this step, you don't need that.
Makes sense.
Yeah. Other questions about this? Okay. And right now, so you can take your migration array that has all your models and just say, plot migration array bracket bracket 17, and you'll see this plot for that 17th model, which is a lot easier than reading the the various arrays we have to say, oh, of course that's that model. Now we can do that, but it's still painful. Okay. So here you see the growth of models, right? And so as mixed by the number of maximum free parameters. And so you can see when you have two populations, you can't get to very high numbers of free parameters, right? Whereas with 3 and with 4, it keeps growing and growing and growing, right? So, 100,000 models is here, right, with 4 free parameters, okay? You're probably not going to want to do 100,000 models, right? So you have to filter this, okay? And here let's look at just 3 populations with 5 free parameters, 5 or fewer free parameters. You can see these are the distribution of models. Okay, and you can see how the models differ in like number of collapse parameters, right, which is the green. Right, so some have no collapses, so like island models, and some have 2 collapses, like a tree model. Okay, you can differ in number of ends you have. You can differ in number of migration rates you have. Okay, so here's all the models plotted that way. Okay, and so this model here has no collapse parameters. It's just an island model with this migration rates. This model here has 1 collapse parameter. This one, how long ago did this population merge happen, right? And then these different migration parameters and so forth. Okay, so you have this whole set of models. Okay, filtering. Okay. So with model tests, if you wanted to, you could choose to ignore models that have both invariant sites and gamma, right? Stomacoccus would be happy if you did that, right? I think those are good models. You can do the same sort of filtering here, right? And we have some functions for filtering, or you can just use information about the models themselves. So we have ways where you can say, here's a list of all the models, show me which models have fewer than 2 free parameters, which models have at least 2 migration rates, and you can get that out. Using the old Laplace function that everyone loves in R. Okay. And now the analysis. Okay. How do you calculate the AIC scores? Okay. And again, these 2 steps, if you don't like using likelihood, fine. You can create your set of models here and then use that to inform some other analysis. Right? So you can decouple those parts. Also, you can create these models on one computer, and then when you're doing the analysis, you can split up across the cluster or something like that. Okay. All right, so how do you analyze this? So get the exact likelihood, right? So I give you a species tree, give you a gene tree. What's the likelihood of that gene tree given that species tree? Okay, and there's ways to do that. Okay, some issues with that. So that might work only for certain kinds of models. Maybe you can't have migration. Maybe you can't have changing population size. Okay, maybe just the math is done for those. Okay, and sometimes also, like some initial versions of COL could take 20 hours to calculate likelihood of a single gene tree. Right? It only takes us 8 hours. So it might be faster to do it for, with simulation. Okay. In general, though, if you have a nice algorithm for calculating the likelihood directly, that'd be faster. That's one thing we're talking about with phrapl is in those cases where there is a fast way to calculate the likelihood, instead of doing simulation, we can just use that instead and pull that in, which will make it faster in general. So the only things you have to simulate for are hard models. Right? Right now, you just simulate for every model. Okay. Just easier first. Any questions about that? Okay. So now let's do some programming. To understand approximate Bayesian computation, right? You can think about it as a simple algorithm. Okay, what we're going to do is show you this, and then we'll convert this into approximate likelihoods. You can see how they relate to each other. Right? So the approximate Bayesian computation for many reps, right? You sample parameters from a prior, right? You simulate data using those parameters. Okay. Then you calculate some summary of that data set, right? Which could be number of DP coalescences, could be an aspect to genus D, could be any number of measures or multiple measures. You compare that summary stat with your observed data summary stat, right? If it's below some magic threshold, epsilon, you say, okay, they're close enough, it counts as a match, right? And then we store that set of parameters, okay? And then repeat, okay? So those sorts of parameters that generate data that don't look like your observed data, we throw out. Those that do, we keep, okay? And when you're done, you can do a plot and say, okay, look, here's our posterior probability distribution of those parameters. Okay. Now, some issues with this is the prior and also this epsilon. Right? How much you distinguish between models can depend on what your epsilon value is. How close is close enough? Okay. So the paper from result from Robert et al. Right, and here we see how often an ABC chooses the wrong model, right, based on these other settings you can put into it. Okay, and finally, use of priors itself, right? I mean, phylogeography matters, right? So if you say there are 2 different species here, and so you can't build this factory here, and so they're not going to have all those jobs to the auto plant, people are going to lose, not be able to feed their families as a result of that, right? Or you're going to lose a species as a result of saying they're one species, right? So your conclusions matter in this field. And with prior, you're saying, okay, I'm bringing my prior beliefs into it, right? And getting this result. It's very easy to be criticized, and it's like, oh, I have different prior beliefs. I think that everything can be lumped into one species. Let's put that in and see how it affects things, right? As we often don't think about priors, you always just use whatever the default priors are for the program or something. But priors can definitely affect results. Okay, we're finding this in my lab. We're looking at beast, which is out of this domain, and finding that priors on branch lengths can have a strong effect on what you get out, right? And so it's not always safe to assume that whatever the programmer thought was a good default for all data sets works well for your data set, right? And so it would be nice if we could get away from the priors. Okay. Now, these are some of the nice things. Bayesian approaches have some nice advantages too, right? It gives you this posterior probabilities and things like that. But this is nice. But what we're doing here is a likelihood and sort of an alternate approach. You can use both if you want. Okay. So approximate likelihood is very similar to approximate Bayesian computation. Okay. We again estimate the data, probability of the data using counting exact matches. Okay. But now we're doing it with exact matches, not approximate. Right. So let's modify our ABC code to be approximate likelihood code. Right. So for many reps, so we move sample parameters up here. Okay. So I say, for a given migration rate, okay, 0.1. Okay. I'm going to, and I do it from a guess, not from a prior. Okay. I'm going to simulate a data set using that. Right. And now, instead of doing a summary stat, right, where it has to be within epsilon, my sort of equivalent summary stat is just topology. And I say, is this simulated gene tree topology exactly equal to my observed gene tree topology? If so, it says that that data set has that probability of generating that gene tree topology. Okay? So now I don't have to get rid of the epsilon thing. We just have exact matches. Okay, because we have this discrete parameter topology. Okay. And now, if that happens, we count it as a match. Okay. And then what we do to calculate likelihood is just the probability of getting our exact data given those parameters. Okay. So this is likelihood. Okay. What makes it approximate is we have to do these simulations things. Rather than calculating it directly, if the probability is 0.1, we do it and find the probability is 0.1 out of 1,000 reps is 10 matches. Well, it could have been 11, it could have been 9, right? So it has a little uncertainty. So the more you simulate, the more precision it has, right? So that's the approximate part, right? But if you're willing to simulate a long time, it converges to be exactly equal to the actual likelihood. Okay, so there's the number of matches over the number of reps you do. Okay, and so cleaning up the stuff I've crossed out, right? That's approximate likelihood. Okay, that's all we're doing. Okay. Just simulating a whole lot of times under a given parameter set, seeing how often we get the observed data. That's likelihood of the data for those parameters. Try a different set of parameters, repeat. That's how you get the likelihoods. Okay. Let's go to coin flipping. Okay, so here's actual coins from Britain that have Darwin on them. Okay, and we say, okay, here's my observed set of flips. Okay. Now if I'm doing ABC, I might do a summary stat. Right? I'm not going to do a summary stat. I'm going to do the exact match. Right, so do many sims, and I say, okay, how many match exactly? And if I do a summary, I could do this way, right? Or exact match, I do this way. Right, so summary can be more efficient here, but we're still doing exact matches. Okay. And so that's the approximate likelihood approach. And so comparisons with ABC. Okay. ABC, we have summary stats. Here we just have topologies. Okay. We do simulation for both. Approximate match here, exact match here. Here we pull from priors. Here we're trying to optimize numerical values, just try a bunch of ones and find the ones that work best. Here we integrate over uncertainty. Here we just get the maximum. Okay. How does this work in practice? So let's say here's my model. Okay. And I have these numerical values for the parameters. Okay, migration rate of 0.2, migration rate of 0.6. Okay. I use MS. and simulate. Right, there we go. So how many people have used MS directly? Okay, many of you. Okay. And so you can see I'm going to generate 100,000 trees, so 100,000. I have 5 populations with 3 populations with 5 samples each, so 15 samples, right? And I tell about how many samples are in each population, and I tell about when I have coalescences, what my migration rates are, and so forth. Okay, simulate a whole bunch of trees. Okay, and then I find that, say, this tree matches my observed tree. Okay, and this step here takes like 5 seconds. Okay. So basically just repeat that many times with different sets of parameters and find the ones that match best given this overall model, and that's the likelihood of the model. Okay? And then I can try a different model and do the same thing. Okay? And once I have likelihood of some for parameters gives me the AIC score, so now I get AICs, and I compare models based on AIC. All right? So this sounds like a dumb idea, right? For many reasons, right? One is tree space is large, right? So here's exponential growth. Whoa, it's crazy. Yeah, it's doubling each size, right? It's on a log scale, obviously, right? 10-fold increase each step, right? The growth of number of trees, double factorial, faster than exponential growth. Okay, rather than doing 2 times 2 times 2, with number of trees it goes 3 times 7 times 9 times 11. So it shoots up really high, right? So for 40 samples, right, there are more trees than the number of atoms in the universe. Okay, so you're going to try to match, exactly the trees with, what's probably getting exactly the same configuration of atoms in the universe each time. Well, you can do that, but it'll take a while to simulate that, right? It'll take many, many samples to simulate. So why do you have any hope of succeeding? Well, first of all, tree probabilities are not uniform, right? So for this particular model, right, if I just sampled all possible topologies from uniform distribution, each topology is an equal chance, I get this line, right? Sometimes I get a couple of copies, but most times I get 1 copy, okay? Whereas if I actually simulate, some trees are present, 30% of the time, right? So I have a good chance of finding those trees in my sample. So if I simulated, 1,000 trees, I might get those, get 30 trees from that, okay? So guess I'm getting biggish numbers. We also have some ways to make it easier to do matches. Okay, so one thing is, I don't really care. So one assumption of phrapl is that within a population, your label doesn't matter, right? So this is go for A from this population for this gene, and this is gene go for B. I don't care about that labeling, right? So we're going to try to match that labeling exactly. What we do is try all possible shufflings of the labelings on the gene trees, right? And then correct for that. Okay. So out of that shuffling, only 1 matches. Fine, but it allows me to find out that frequency of those matches. Okay. So then it makes it a lot easier to get a match. Okay. But then we can back correct for that. That's one efficiency we have. And so we actually, when you run phrapl, you'll see it first generates all these weights for gene trees, which comes from basically this approach. Okay, so we can correct for that. We can deal with polytomies. So maybe your data is perfect and you have nice, well-resolved gene trees all the time. Okay, probably not. That's what you do in that case. So what we do is allow for polytomy resolution. Okay, and so here there's 3 ways to resolve this polytomy. Only one of them matches here. So we count that as 1/3 of a match. Okay, so if there's polytomies, you can correct for that. And if you have polytomies that have higher degree, polytomies with 4 taxa or 5 taxa, we correct for that appropriately as well. Okay. These ideas make sense? Yeah.
Does it force polytomies to be soft?
Yes. Well, no, that's an option. So if you can pass it as an option and say, I only care about exact matches. But yeah. Why would you want? Why do you ask?
I mean, what if I have a true hard polytomy? I mean, I don't, but
yeah. Yeah.
Okay.
Yeah.
All right.
Yeah. Other questions about this? All right. Another idea, and this is something that Nathan suggested that I was resistant to for a while, I'm still sort of, is subsampling. Okay. It works, so I should shut up. But, you're taking your data and you're saying, okay, tree space is huge. I have 20 samples from this species, from this population, and then 3, 3, 3. Do we need to have all 20 to get the right model? No, it could probably make do with 3 or 4. Okay, and so we have ways now to subsample down. Okay, and then cross out B2, B3, and then see how many matches I have in that case. So now I'm going from think of the tree space increasing double factorially, right? By dropping the number of taxa, I'm making it much easier to get a match. Okay. Now the problem with this is you've hacked through the jungle to get that sample, right? And you've paid $1,000 for the sample, and I say, okay, we'll just toss it. Right? So as an empiricist, you're probably resistant to that. But we found that you can actually estimate the proper model correctly by doing this approach. It also saves you a lot of time. Okay. And then something we're still playing with. The best way to do the subsampling is a way to, if you have more data, is a way of sort of rewarding you for more data by having AAC weights different, having more definitiveness. And so we're still playing with that, but right now subsampling seems to be working. Any questions about that? As users, are you happy to allow subsampling? I said to you, I can do a run in 10 hours of subsampling or 1,000 hours without subsampling. Would you be willing to subsample your data and throw out stuff, or would you?
Preliminary exploratory purposes, it'd be nice.
Yeah. For publication, though, would you? Because you could just the truck could have broken down that day. You might not have gotten that specimen, right?
You're not necessarily throwing out data. You're just like looking at it in small, discrete chunks.
That's true. I mean, we use subsample with replacement. It's not that that sample is never included. It's we include that sample and then draw another 3 from that population, another 3 from that population. So if there's one that's sort of weird in some way or giving some other signal, we pick that up. So you're right. It's not just throwing it out.
You can sample enough times where you get pretty good confidence that you're sampling all of your samples at some point. They're just not all at once.
How much of a loss in precision do you have?
So we haven't detected, we haven't looked close enough to know about the loss of precision. One thing you do possibly get is a lower magnitude differences in AAC scores and lower differences in relative support for different models. And so one thing we found actually early on that's kind of cool was that if I have 3, 4, and 5 samples per population, and look at AIC, right? I would find, this and this, and then if I, do linear regression, just 2 points. So with any fewer, I'd find that the actual number for 5 was close to that. And so I would try a different model. And again, we do the test here and find it matched pretty well. And so what we were hoping was that, okay, now you've gone and you have, 20 samples. We can do the subsampling for 3 and 4, extrapolate, and then find we have these big AAC differences, right? And that doesn't seem to be holding with empirical data sets. We have to investigate that more. But that sort of thing would allow you to, you have a ton of data, so you should be able to differentiate models well, big delta AACs, right? And if the subsampling is a good estimator of that, then it should work. But it doesn't quite work yet, so we have to figure out that more. Yeah. Other questions about this?
I think it's good to have the option there. Potentially, I suppose you could see it as some sort of simulation. Use a simulation and plug that into phrapl for some sort of study design. Some sort of estimate of how many samples do I need, per location, across a range.
I didn't go that way, yeah.
What does that do then to the inferences that you draw from phrapl?
That's true. One thing you could do is use the same machinery and say, okay, I actually have 5. Let me go back to 4 and 3 and see if it gets the same model, relative model support. Maybe I don't have to do all that. That's true. Yeah.
So, but I think one thing Nathan's point is important to recognize. It's not as if you're not using all of the data. So for most phylogeographic studies, we have a number of samples such that, if you think back to this figure that Brian had about the number of possible trees. So the number of genealogies is so huge that it's really hard to calculate these likelihoods if you have to rely on simulating genealogies that match the pattern you see in your data, right? It's common for us to have hundreds of samples for phylogeography, which means that there are many, many times more genealogies than we could ever possibly simulate in any infinite amount of time. And so the coalescent model assumes that you're drawing samples and that the alleles that you're sampling from the population are representative. And so in a sense, when you do subsampling, you're just taking a smaller sample from your population and repeating that smaller sample some number of times. And so the reason that it's advantageous is that if you have a fewer possible number of genealogies in your sample, then you will have a better chance of figuring out if you have a match to those genealogies for a particular model. And so it seems like cheating and like you're not getting as much information out of your data, but if you sort of step back for a second, you actually are increasing your ability to get information out of the data by looking at it in more discrete, smaller units, over some number of replicated times.
Sure, and also when you think about estimating likelihoods, right? So I would rather, get a better estimate by doing 30 matches out of 1,000 samples than one match out of 1,000 samples, right? Because one's very similar to 0 and 2, which are very different in likelihood, right? But 30 versus 29 or 31, there's more precision there, right? So by, if the actual probability of the gene tree gets really, really low, you just need many, many more to get up to 30 hits. Yeah.
Do you run into bias by using this sort of, I'd assume you would, depending upon what you picked as your representative to be.
We loop over them. So we don't just pick, it's not always Bob. It's sometimes Bob, sometimes Mary. So we do this multiple times. And so you sample, using all the individuals you sample with equal frequency. Yeah. Because you're right, that could cause a problem.
I mean, another thing to remember too is when we sample alleles from a population, we find that some individuals have the same allele a lot of the time because that allele is really common, right? And so when you do subsampling, you're more likely to sample those common alleles than the run that you only find in a single individual. And presumably, if you have a larger sample, even when you do subsample, you're going to most of the time sample those alleles that many individuals have, and only rarely sample that singleton that you just see in one individual. So.
Other questions about this? Okay. Another clever idea, clever property, is that when you do many loci, so oftentimes in phylogenetic stuff, as you add data, the problem becomes worse exponentially or worse than exponentially or something like that. Here, the amount of time it takes to compute it grows slower than linearly, right? So rather than just one, it's not just one-to-one. So if I add, if I have 5 genes and add 5 more genes, it doesn't take twice as long. It might take 1.9 times as long or something like that. Because we have the same time to simulate the gene trees, the same MS time is the same for 5, 10, or a billion gene trees, right? So we still have the time to do the matching. Right, but also we can save on that too by, if I have the same gene sampled multiple times, I can just count that twice or 3 times. So I have savings that way. So the good thing about this is as we scale up to genomic data sets, right, we have lots more information, but the runtime scales even better than linearly with that. Okay, so it should be as feasible as it is now, right? Which is a nice property. And also, maybe you have another clever idea about this, right? Because you're our guinea pigs, right? So think about what you want, but also if you think of any good things to improve, you're open to that. Okay. So now this analysis step. So now I know how to calculate the likelihood and how to do that efficiently. Okay. And now to think about, how do we get these parameter values? Right, because I have this particular model, and I choose really bad parameter values, I'll say that model is a terrible model. Right? Look at anything else. If I say you're doing a GTR plus I model for phylogenetics and I say I is 0.9, it's actually 0.1. Right? That model is a very bad fit because I have a bad parameter value. How do we get these numerical parameter values for this given model? And there's two possible approaches here, right? So here's my numerical parameters. And let's imagine I have a 2D space, so I only care about 2 parameters, right? And this is some sort of peak here, right? So I want to find the best parameters are here. One approach is a typical numerical optimization, right? Try a value, okay? Try another value, is it better? Yes, okay. Next value, try is it better? Climb the hill, okay? And that's how we did this initially, and there's still code in there to do it this way. But Nathan did a very clever thing and actually looked at what was happening, but what values the program is trying as it goes. And what he found was that for certain parameter values, it would just find one just to get stuck there and change the other parameter values. This parameter value would be pretty bad. Okay, so what we now do instead is a grid search. So you say, okay, here's where the bounds come in. So, give me a range of possible migration rates. Give me a range of possible taus. Okay, and then we'll try all combinations here. Okay, so of course, the more parameters you try, the more time it takes, the better chance you have of getting the right estimate. Okay, and you find this works a lot better than actually numerical optimization for the same amount of time investment at getting the best values. Okay, and so you find, here, the best value is like right here, but this is also probably pretty good too. Okay, it's not quite at the optimum, it's not quite here, but it could be close enough to do this. And so again, what you could do, and we have this built in, is a 2-step process where you find the best ones and then search more closely right there. Okay? Improve it that way. One nice side effect of this is that rather than getting, this is the best one, you get, this is the best one, but also you know something about the contour around there, right? So you can get some sort of confidence about, how much uncertainty we have in this estimate of time, which is good too. Okay. Any questions about that? Yeah.
When you're doing this grid-like search, because you're doing like an MCC sort of deal with this. Is that how this? No. Oh, you're just trying to find the optimum.
Yeah, just trying to find the optimum.
Well, does it matter? So in the last one, the more times you run it, the more likely everything's going to cluster in that one optimum.
Yep.
So with this, do you just run it once, or you run it a few times, or you just make the grid smaller and smaller so you still get a nice heat map?
Yeah. So the 3 things you can do. If you can run it once and then do a finer grid here.
Okay.
You can run it if you find that your best value is at the edge. You can then make a new grid around that point.
Okay.
And make it even further out. And then we haven't done a lot with visualization for this, but one thing you can do is the Akima package to you do a contour map without a uniform grid spacing. So if I have tons of points here and scattered points everywhere else, most contour mapping won't work that way, right? But with Akima, you can interpolate evenly, and so you can get good contour map there. And actually in the lab, we have another package that can do multidimensional interpolation. Because right now R can only do 2D, but in theory you can do multiple D. If you're patient.
The number of dimensions here is equal to the number of parameters.
Yeah.
Okay.
Yeah. And we have bounds, so migration rates below, I think 0.05 is it?
Yeah, 0.05. Yeah. 0.05 for migration.
So the question, if you're trying to compare models, right? So one model has a migration rate of 0, one has a migration rate of 0.000001, right? Generally spending time on trying to figure out if those are different or not. Probably not. So maybe we'll just set the min bound at 0.05. So some migration, but not a ton, right? So if you disagree with that, you can change those bounds. Okay, it's all made to be very flexible, so you can go in and fix that if you want. Yeah. Other questions about the grid? Okay. So we have 2 ways of then optimizing across model space. So one way is exhaustive, right? So for every model, you find the best numerical parameter values and then calculate the AIC score under those parameter values. Okay, that's the way I probably recommend in general, right? Because that way it tells you, here's the best model, here's the next best model, here's the third best model, how different are they? I can do model averaging. I want to say, okay, what's the average migration rate between these 2 populations? I can do it under this model, which has 90% of the weight, and this model has 10% of the weight, and not, fix my cart just one model. I can sort of integrate over that uncertainty by doing it that way. Okay, so I'd recommend that. Okay, If model space is huge, though, maybe you can't do that. Same way, we're looking at trees, like just regular phylogenetic tree inference. Ideally, you could say, okay, for every possible tree, what's the likelihood when we look at that confidence interval of trees? In practice, you can't. You have to do heuristic. So we have a heuristic built into phrapl, a genetic algorithm search. Okay, and so in phrapl, you'll see migration array stores all the models. There's something called a migration array map that stores model similarities. Okay, and so what you do is you have a set of possible models. Okay, and for each one you optimize the continuous parameters, right? And then you find out the AICs for them. Okay, and then we can throw away the ones that are bad, and then do some sort of swapping between these. Okay, so genetic algorithm is actually evolution happening. So each model is a little critter, right? And you find the models that are the fittest, and they get to have sex and recombine, right? And they make new critters, and then we keep running it. That's sort of algorithm. And so the migration array map that we require you to make is a way of saying how similar are these models, and how can we switch model from model to model and have them recombine. Okay, but again, for most users, I would just recommend doing the exhaustive search. Okay, and exhaustive search is one of those problems that computer scientists call embarrassingly parallel, right? Which means that things don't have to talk to each other to work, right? So think about like a Mr. Bayes MCMC run, right? They have these cold chains and hot chains, and periodically they have to swap back and forth, right? So they'd be able to talk to each other. Right? Here, they don't have to talk. I can say models 1 through 5 go on this computer, models 6 through 10 go on that computer, run. Okay? And then when they're all done, I can bring them back together. But while running, they don't have to talk to each other at all. Okay? So it makes it very easy to parallelize at that stage. Is there a question? No. Other questions about this? Okay. So, and R has very good ways to parallelize now. So you can use MC-Lapply. So if you have your computer that has 4 cores, you can use MC-Lapply, apply this to all your models, and it'll run them all in parallel. Okay, on your desktop, you can run it on 12 cores. Or you can send it out and use actual batch scheduling software on a cluster for you. Okay. The underlying structure. So most of it is R. It's an R package. Okay. Some of it is Perl. Okay. And the reason for this is all the basic stuff was done before grant deadlines. I could do Perl quickly and do matching quickly. Okay, and I've gone back and made it a lot more professional, work a lot better, but we found that when we moved this particular code into R, it made it run a lot more slowly. We said, okay, you know what? It's a pain to install Perl perhaps, but you do it once and then everything runs faster afterwards. So for right now, we still have this Perl dependency. And of course, you still also have MS, so dependency for the simulation. Okay, so here's a few empirical examples. We'll talk more about examples tomorrow. Okay. So Salter et al. Just studied these spiders, right, and found these 3 populations. Okay, and we ran it through phrapl. And the initial models were all, these closed models that have no migration, right? And we found, and so, it makes sense, you use these approaches for species to limb. They don't allow migration, right? Maybe migration is happening, right? So we throw it into phrapl, whole set of models that range from the species no migration models to island models, and found that actually the best model in this case was an island model, with a lot of weight. Okay, so now we discovered something new about those data from using this exhaustive approach. Okay, and then what you can do is say, okay, that's great. Now phrapl gives me these rough estimates of parameters. Maybe I want to go and run it more intensively, get better estimates. Or maybe I say, okay, you know what? At that point, let's use migrate or something else that does a better job, more efficient, and bring the model into there. So that's a possibility too. Here's another example where we looked at some of Brian's meiotis data set. It's meiotis, right? Mm-hmm. Yeah. You can tell from the canine structure. Where there are different possible models that could run. And the nice thing is, in traditional approaches, it might be hard to compare a star-beast run from a migrate run, which model is best, right? Because they're different criteria. But phrapl, we put them all in the same thing. We can do all those models, right? And find that, this model is the best model, right, that has some population coalescence and then some migration rates, right? Whereas this model is the second best model. It's fairly worse. Okay, so it allows you to make them, and so you can say, are they still exchanging gene flow? And this second best model shows, no, they're not anymore, right? The model that's a little better says that they are. So you go and say, okay, let's look at these models in more depth. Maybe we can look at other data. Right? But it tells us we have information about this. Okay? And I'll explain why we sort of had different results from different approaches before. Right? Some that allowed migration, some that didn't have migration. Okay? So it allows you to explore this model space. So that's basically how phrapl works. So the high points are it's a way to create many, many models, right? With some parameters, with some limits to it. Allows you to evaluate many models. Okay, it's prior free, right? So you can't skew your results to pave a park to pave a wetland or not pave a wetland, or anything like that. And it should scale well with a number of gene trees. Okay, so it will scale well for next-gen approaches. All right, so that's the basic structure of how it works. And we'll run on some example data sets soon. You can see how it works in practice. And tomorrow we're going to talk about some analyses where we show, with simulations, how well does it recover the true known value, and then how well it works across different data sets too. Okay, and of course we got money from people. Yay, money. Yeah. So the questions about this. Other questions about this? Yeah.
Just out of curiosity, how many loci have you ran it with so far?
The most I've done personally is, I think, 5. How many of you have done?
I mean, with simulated data, I've done on like 100.
Most of the empirical data sets we've run have been sort of the 5 to 10 Sanger loci sorts of biogeographic data. So we haven't used big next-gen style data sets yet. But I mean, I know Ariadna is working on developing one of those for her dissertation project. I know many of you are in the same position. And so again, there's no reason to think that it will work, and it probably won't take that much longer, because the big computational effort is simulating the models and the gene tree distributions in the models. And then if you have 1 or 100 gene trees, that's not as much of a problem.
How would phrapl handle one locus being like a tooth comb and then another one being highly structured like site B?
Yeah, I mean, so it can deal with it can allow soft polytomies. Whether that bush would actually give you any information, I don't think it would. I mean, it shouldn't. In theory, it tells you some information about like, oh, it's recent and stuff like that. We don't really use branch lengths. So that probably doesn't help much.
All right.
You would hope.
So talk a little bit about how we calculate the AIC across loci. So if we have a match, out of 100,000 simulated gene trees, it matches 10 times in locus 1 and only 1 time in locus 2.
Yeah.
I mean, that's a good way to think of it.
Yep. So it's just like how we calculate likelihood with DNA, right? So with DNA, if I have this and I have AATT and I have AGGA, right? The likelihood of this data set is the likelihood of this. Like 1 times like 2, or log like 1 plus log like 2. Right, so that's what we do for DNA. What we do for phrapl is we have for that species tree, I have gene tree 1, right? And I have gene tree 2, right? And I have this matches 5 out of, 1,000,000 times. And this matches 2 out of 1,000,000 times, right? That's my likelihood, right? And so you just multiply the likelihoods or add the log likelihoods. And that's the likelihood for this thing, right? And so what I would find is if I have something that has no information about any of the species trees, that I have a different species tree. Something like that, right? Then this might also, so that's species tree 1, species tree 2. I might also still get 5 out of 1e6, but here I might get 20 out of 1e6. Make sense? And that's likelihood, and then just throw in K and double it, and you have AIC. Yeah.
I have a naive question. So, how would that work with for comparative phylogeography? I mean, instead of using many loci for the same species, you have like different species that you might have a reason to think that they might share a common history.
Yeah. So that's fine, right? So with phrapl, so here, imagine we have, some migration here, and here no migration. So A, B, C, you might say A and B B and C might be one species or different species. I'm not sure, but they're definitely different species from A. It's also a species tree or a population history. So it's fine. It doesn't care whether it's species or not.
No, I mean, if you have like, if you're planning to do a comparative phylogeography in the sense that you're using different organisms.
Oh, okay. Sorry.
And I wonder how the model selection would work, and because it seems that, I mean, intuitively, if you have organisms that don't share the same history, phrapl will have problem finding a model that has a higher probability.
So that would be a sort of a higher level loop than this, right? So what you could do is run phrapl for each organism, right? So here are the crabs of this area, here are the birds of this area. So you run phrapl for each of those. And what you could do is link the parts of the comparative phylogeography you care about. So I say, okay, I don't care whether the birds and the crabs differ in this migration rate, but I do want to constrain them to have the same overall population history of coalescing. You could do that. Yeah, so what you would do is, run phrapl for each of those things independently, and then sort of match up the models and calculate the joint AIC of that set of models. And that would work. It's something we've envisioned, but that's a very cool extension.
Okay.
Yeah.
How do you deal with issues where the gene trees don't match the species trees due to maybe hybridization or something like that? I mean,
right. So if there's hybridization within the gene trees, we don't deal with that yet. So a lot of work in coalescence recently has been about have this one gene evolving, but have this segment comes from this history, this segment comes from this history, and they come back together. We don't deal with that yet. In theory, you could slap in a different simulator that deals with that and use that. So I'm not sure. I think there's some MS versions now that deal with hybridization, recombination. And so if that were happening, you could use that. But then again, think about the model complexity that comes as a result of that, right? So you have this recombination parameter. But in theory, you could do the same basic approach. Is that something that matters for your data?
Yeah, it's something that I'm kind of interested in.
Yeah. Yeah, so I think it could be a pretty easy extension for this. Other than like the runtime stuff. Yeah. Computers are getting faster all the time. You just throw money at it. Yeah.
So, do you suggest using IMGC, some kind of software that will remove recombination from your data sets before building the trees?
That probably makes sense. I mean, there might be a bias that comes from throwing out those data sets rather than others. I don't know. What's your sense?
So I guess my advice would be we want a good estimate of a gene tree that fits the standard neutral assumptions of the coalescent model. And so just like you would with IM or migrate, you might want to clean your data beforehand to, if you have evidence of recombination at site 800 out of 1,000 base locus, you might just analyze the first 800 bases in that case. You probably would do the same thing here. I think that any reason you find selection or you think there's selection, that would probably be a good argument not to use that locus because it would violate the assumptions of the model. Yeah. All of those standard caveats would apply in this case.
I mean, I could still use a bias if like all loci are recombining, but you can only detect certain ones that have a certain pattern as recombining, the others you can't. Then those ones that you're still in might have been through a recombination history, but they're biased in some way that you can't detect that. And so you could get the wrong answer. So if you think it's a few bad ones, toss them. If you think your overall the model's wrong, then change the model.
Well, and so I think probably what it would do is if you have recombination that you ignore or you don't detect, that's going to make the topological pattern a very poor match to any of the models because it's not a genealogy that would evolve under a neutral coalescent anyway. So it probably won't mislead you. It might just decrease your ability to differentiate the models. You know, if that's one locus out of 100, it's probably not going to be a big deal. If it's 1 out of 2, it might be. Didn't you have the obligatory picture of your kid on the test?
I might send it to put down like, you know, like